Okay. And so like I said, all of this will be up on Schoology by the end of today. So when we're looking at active transport, here is our definition right here. It is the movement of particles from now areas of low to high concentration. So this is the opposite of when we were looking at passive transport. That means that instead of going down the concentration gradient, we're really establishing or making a concentration gradient. Yellow? Not a good choice. Okay, so here's what I want you to picture. Does anyone have a basement? Right? Okay, so here is the ground. Here's your basement down here. Here's your house up here. And what is one of the worst parts about having a basement potentially?
Spiders.
Yeah. Okay, it's cold. There's spiders. Okay, so when it rains, the water table rises, and some of that water can get into your basement, right? Going from an area that is higher to lower. We can picture this. That's easy enough. Do you have to put energy into it for that to happen? It just kind of does it, right? What do you do then to get the water out? You have to pump it out. What do you use? Sump pump. So you get your sump pump or your shop vac or whatever, and what do you need to be able to make that sump pump work? So if you lose power, all of a sudden, now your basement's flooded, and if you don't have a generator, you don't have a way to get this water pumped back up. So active transport's the exact same kind of idea. We're going to take molecules that are, so here's our membrane here. We've got one side with a high concentration, and just based on how molecules move, they're going to want to go this way. However, instead of them crossing the membrane that way, if there's a low concentration over here, and these are molecules that the cell needs, then we're going to have it come on back this way. I know what they're here for. Literally seconds after. Here, let me pause this. Okay. So instead of moving molecules down their concentration gradient like they're going to want to do, if they're molecules that a cell needs to have inside, but there's already a higher concentration, or if it's molecules that the cell needs to get out, even though there's a higher concentration outside, we can use pumps. So let's quickly kind of compare the difference between passive and active transport. If we take a look here, on the left, what are we showing? These three images that are all on the left.
Passive.
Where do we have a higher concentration of molecules? On the top. On all three of these, we have a high concentration up here. What do we have on the bottom? Lower concentration. We have molecules that are moving across the membrane from high to low, so that must mean that it is passive, right? What's going on in this image right here? It's going directly through what? There it is, the phospholipid bilayer. And therefore, this is diffusion or simple diffusion. And both of these are facilitated diffusion. Okay. The difference here is that in, in active transport, we're using energy to move molecules in the opposite direction of how they naturally want to. So where do we have a higher concentration of rectangles? On the bottom. They're getting pumped up. Where do we have a higher concentration of circles? On the top. They're getting pumped. Wait, I said that backwards, didn't I? Yes. Higher concentration of the squares down here. These are the ones that are getting pumped down from where there's fewer to now where there are more. Same thing's going on in the opposite direction with the circles. And what is this little guy, right there representing? Energy in the form of?
Sugar. Glucose.
ATP. You were right. ATP. So we have to use energy to make this process happen, just like it happens in your house. Questions so far? Okay. Yes.
What's the difference between the facilitated diffusion and so there's passive diffusion?
Passive. I'll give you kind of a rundown of all of it towards the end to make sense of it. No, let me do that now. Okay. So I'll come back to this here in a little bit. So when we're talking about membrane transport, What are the two types of membrane transport that we've now looked at? We've now looked at active, passive. Tell me some things about passive transport. 3 things that y'all should know. No energy, high to low, until hold that thought. Okay, so we're reaching equilibrium.
Can I regulate?
Yes. Okay. What have I now said about active transport? Requires energy. Molecules are moving from low to high. And it establishes a concentration gradient. I'm going to really abbreviate here. Okay? All right. Types of passive transport that we've looked at. It can either molecules can either move through the phospholipid bilayer or through some type of channel. So what are the 2 types that we've looked at? So what are those 2 types called? When it goes directly through the phospholipid bilayer, what is it? Simple diffusion, and if it's not simple, it is facilitated diffusion. And a type of facilitated diffusion that we looked at is osmosis. Okay? Raise your hand if you're solid there. Got me a little nervous. Okay. So now we're looking here. We're looking at active transport. We're looking at how the cells are able to move material against their concentration gradient. And there's 2 ways that the cells are going to be able to do those. Either by using what are called pumps, which is a type of molecular transport. We're going to go with yellow. Or bulk transport that gets a little bit more tricky. Pumps are really easy to picture. If you've ever seen a sump pump, if you've ever heard the word pump, that's it. That's all it's doing. So we've got these proteins. This integral protein that goes through the membrane that is taking molecules from the side where there's a lower concentration to where there's a higher concentration, and that is going to require the use of ATP. Easy? Why would you do this? Turns out it's super duper important in one type of body cell in particular, and that's going to be your neurons. I do not expect you to memorize any of what I'm about to say other than understand the fact that sometimes some cells have to have this huge concentration difference. In this case, this is the inside of a neuron, and this is the outside. Did anybody read from the text or scan through? Okay, what can you tell me about your neurons? They're tiny, they fire, they take 1/4 to 1/3 of the energy that you use every day just to use these pumps. So these pumps require 1/3 or so of all the calories that you eat every day. You're eating it just to be able to have nerves be ready to fire. And what are they doing? They're pumping potassium in to make a really high concentration of potassium on the inside, and they're pumping sodium out so that you have a really high concentration of sodium on the outside. If you don't have that difference, you can't fire your nerves. So how do batteries work? Does anyone know? Okay, if this is my battery, what should I draw?
You need a plus side and a minus side.
Plus side and a minus side. What else am I going to need?
The little tips.
That little nubbin, right?
Yeah, that's
Okay, that plus side and that minus side, I didn't know if anyone would get that one. That was kind of what I was hoping for. And they're separated in the middle. What makes a battery is that you have more electrons on the minus side and an overall positive charge on the other. It is that difference that lets the battery work. What happens if there is no difference? It's a dead battery. Once you've reached equilibrium, once there's no longer a difference in charge, your battery's dead. It is exactly the same as your neurons. If you no longer have a concentration difference from the inside to outside, they can no longer be ready to fire. Okay? All right. So those are your pumps. Merci beaucoup. Now we need to look at a different type of transport. I'm going to pause. Okay. So we talked about pumps. Now we're going to look at bulk transport. To understand bulk transport, we need to revisit membranes, where they are found, and in specific, what type of organelles have membranes. Look at this picture and see if you recognize any of these parts here. Okay. So on the top left, does anyone know what that is? Say it out. What is it? Someone give it to me. Okay. So up here we have the ER. Remind me again what the ER does. Okay, so we're getting this modification of materials. What is this? Blah, blah, blah. This thing right in here. You just say it out if you know it. Ooh, nice. Golgi apparatus. What are the little dots all over the place? Vesicles in this case. Vacuoles are no different. It's just that they're bigger, okay? So plant cells have one large central vacuole, but these vesicles are pretty small. And what are all of these things wrapped in? Membrane, okay? What's important to understand is that membranes can fuse together. So picture a warm, sunny day, because number one, I think all of us need to be in that mental space right now. So it's a warm, sunny day, and you're outside, and you see kids playing, and they look so sweet, right? And they're blowing bubbles. A little wand, and they're going all over the place. Have you ever seen what happens when two bubbles join together? So you get two that are close to each other, and sometimes then you end up with something that looks kind of like this, right? Where there's like this film between the two bubbles. And then that center part's gone, and then you just have one big bubble. Have you seen that? Can you picture that? Easy enough to picture? The membrane acts very similarly in that it can fuse with other membranes, or it can kind of pinch apart from other membranes, just like little bubbles. So if we take a look over here, what's going on is this phospholipid bilayer right here. I should pick one that's not like right where words are. Okay. So this phospholipid bilayer right here, we've got all of our hydrophilic heads up here, hydrophobic tails in the middle. That phospholipid bilayer can kind of pinch out to make a little vesicle. Okay, and those vesicles can reform when it touches another membrane, just like little bubbles. Still so far so good? It's how your cells get material around, right? Package it up, send it somewhere, that vesicle opens up, but it's also really important because it's how you get stuff into or out of your cells. Okay, so we're gonna call these forms bulk transport. We're no longer looking at ways of getting stuff across the membrane, it's getting wrapped in membrane. So let me show you what I mean. Here's 3 examples of one type of bulk transport. So these are both called, what's going on here? Is stuff coming into the cell or going out of the cell? In. What's the term that means into? Endo, nice. So this is called endo, meaning into, cyto, meaning cell, cis, meaning it's a process. So endocytosis. Endocytosis means taking the membrane, wrapping up something that's outside of the cell to make a vesicle that then can be brought into the cell. Do we understand that? Okay. We can do it with food particles. And so because it's like food, it means to eat. Phagocytosis means cell eating.
So at some point, the inside of the bilayer is exposed to water.
No.
How does it just take that stuff and put it in its own little thing without?
I have a video. I'll show you right after. But you're right. It looks like it should, and parts of it does very briefly, which is why they move then to form the way that they do. So I'll show you that, okay? So in phagocytosis, we're going to wrap around something and take it in. Do you guys know of any parts of your body, any cells in your body that take in big chunks of stuff?
Red blood cells and oxygen.
Oxygen's super tiny, but it is a type of blood cell. What's the other type of blood cell? White. And what do white blood cells do?
Bacteria.
It's all about your immune system. This is what they're going to do. They're going to bump right up to a bacteria, wrap the bacteria up, and ingest it. Why would you do that? So that you can kind of puke it back out and show all the other cells, hey, this is the bad guy that I found. That's a whole nother course right there. But this process of phagocytosis, we can look at in terms of our immune system. We can also do what's called pinocytosis, which is where instead of solids, we take in some kind of solution and drink it, right? So this is cell drinking versus cell eating. Okay. So if we can take the membrane, wrap it around something to make a vesicle, is there something that would be the opposite of that? Expelling it. What do you think we could call that? If it's not endocytosis, we could call it. So let's do that. Okay. So exocytosis, that is going to be when we take little vesicles full of some kind of product. They fuse right here. Actually, that answers your question here, so I'll show you that in a second, Dingus. They're going to fuse with the membrane, and then that product can get spit out. For example, your pancreas has a whole bunch of cells that have insulin in them. That insulin has to get excreted using exocytosis. Your neurons do this every time they fire and communicate between neurons. Bless you. So your body is doing all kinds of endo and exocytosis all over the place all the time. Okay? So I told you I'd kind of explain this a little bit better. So if this is one layer of membrane right here, and the tails are going this way, and this is the other layer, and the tails are going this way, they're still protected against interfering with water. Right? And then we've got another layer
so it
here.
never actually like disconnects at any point? Like how do you go from it
And then it will.
breaking off to it being on its own isolated?
I like that question. I'll show you a video on that right after this. Okay?
I mean, it's like it's not that deep. I'm just
No, I think
confused,
it's
like,
helpful to visualize.
the
So
whole
if we,
point is to not touch water on the inside. That's all.
it is. Yeah, we can still do that though. So then where did I put my little diagram here? There it is. Okay. If we come back to this, passive, we talked simple and facilitated diffusion. Active, there are two types. All right, so we've got pumps and bulk transport. When we're looking at pumps, that is stuff actually crossing the membrane. So for all of these, stuff is crossing the membrane. Bulk transport stuff is not actually crossing the membrane. It's either coming in or going out. What are the two types? Coming in is what? Endocytosis.
I'm not gonna remember that second part.
And what is going out?
Exo.
Are there different types of endocytosis? Eating and drinking. Phago, which means to eat, and pinocytosis, which means cell drinking. So when it comes to membrane transport, there it be.